This is the solutions video for problem set 9 for Kevin Ahern's BB450 at Oregon State University. In problem set 9, I asked the students 5 questions. The first question involves, how does a homotropic effector give a sigmoidal V versus S plot, but a heterotropic effector does not? Well, to answer this question, we have to remember, first of all, what a homotropic versus a heterotropic effector is. So a homotropic effector of an allosteric enzyme is a molecule that is a substrate of the enzyme. A heterotropic effector, by contrast, is a molecule that is not a substrate of the enzyme. Now, when we think about how we study the velocity of an enzymatic reaction, we plot V0, which is the velocity of the reaction, compared to the substrate concentration. The substrate concentration is varying throughout the experiment. So we can imagine that as we vary the substrate, we would see something that might be very much like what we saw when we varied the oxygen concentration with respect to the binding of hemoglobin. And in fact, that's exactly what we see. We see a sigmoidal plot where the enzyme is fairly low and active at low substrate concentrations, but as the substrate concentration increases, the enzyme changes from T to R, and we see the activity go up. As we get to high substrate concentrations, the enzyme starts to get saturated with substrate, and we see the sigmoidal plot that we saw very much like we saw in hemoglobin's binding of oxygen. By contrast, when we look at the plot for a heterotropic effector, we remember that, of course, we're varying substrate concentration. We're not varying heterotropic effector concentration. So the heterotropic effector for an enzyme is simply leaving the enzyme in either the T or the R state. It doesn't matter then whether the enzyme will not be doing any flipping. So an enzyme that simply has a heterotropic effector will exhibit the kinetics very much like what we saw of the binding of oxygen by the myoglobin. That is, that there's no flipping of the enzyme or no changing of the enzyme structure during the given concentration that the heterotropic effector has to begin with. So that the only variable, which is the substrate concentration, which in this case is not affecting the enzyme's activity, has only the effect that we see of the normal V versus S hyperbolic plot. The second problem asks a suicide inhibitor of ATCAs called Paula that closely resembles aspartate inhibits the enzyme completely at high concentrations, but at very low concentrations actually stimulates it. Propose a mechanism consistent with this observation. Well, we remember that ATCAs is an enzyme that involves both homotropic and heterotropic effectors. For this problem, the homotropic effect is the most important one. The enzyme, of course, has 12 individual subunits, 6 catalytic subunits, and 6 regulatory subunits. And the catalytic subunits are affected by the substrate, which in this case is aspartate. Now in this problem I've said that the Paula is a molecule that resembles aspartate very much. However, it inhibits it at very high concentrations. Well, what's happening in this reaction? If we think back to what aspartate does, what aspartate is doing is aspartate causes the enzyme to flip from the T to the R state, thereby activating the enzyme. Paula, if it resembles aspartate, likely has the same properties of flipping the enzyme as aspartate did. In high concentrations, we could imagine that where we have 6 catalytic sites, let's say these round ones are the catalytic sites here, that these catalytic sites would all be occupied with Paula. So I put P's here to designate each one. Therefore, if every site were occupied by Paula, the enzyme could flip into the R state, but since it would be bound to Paula, it wouldn't be able to bind to aspartate, and the enzyme would be inactive. However, if we imagine a scenario where Paula was present at low concentrations, we would see that some of these sites that are bound by Paula at high concentration would in fact be bound instead by aspartate. So we might have Paula binding to a couple of the sites, but those couple of sites that are there would cause the enzyme to flip into the R state such that the other remaining sites on the enzyme would be completely active and the enzyme would in fact continue to function. So that's why high concentrations of Paula would stop the enzyme from functioning, but low concentrations would actually stimulate it because it would flip it into the R state. Problem 3 says to contrast the effectiveness of inhibition of enzyme activity by Zymogens versus allosteric effectors. Well, this is something I actually talked a little bit about in class, but suffice it to say that when we think of allosteric effectors of an enzyme work by what I call an on-off switch. And that on-off switch basically involves flipping the enzyme from the T state to the R state, or by flipping the enzyme from the R state to the T state, depending upon whether we activate the enzyme in the first case or we inhibit the enzyme in the second case. Well, both the T and the R states of an enzyme are in fact active. The T state of an enzyme is less active. The R state of an enzyme is more active. So allosteric effectors that flip the enzyme between the T and the R state are really turning the volume up, as I noted in class, or turning the volume down, but they don't completely inhibit an enzyme. On the other hand, Zymogens are enzymes that are synthesized in a completely inactive form. And usually this occurs as a result of the fact that the form of the enzyme that's made originally, that is the inactive form, has its active site covered up by a peptide, by peptides and, that are surrounding it. In order to expose the active site to substrate, it requires a cutting of the enzyme to expose the active site to the substrates. So in the case of Zymogens, we typically see that there is absolutely no activity to begin with because the active site isn't accessible by the substrate. And then after the Zymogen has been activated by cutting a peptide bond and exposing the active site, we see that the enzyme in that case then is completely active. So Zymogens tend to have more of an on-off kind of switch, whereas allosteric enzymes have a volume up or volume down switch. I should also note that Zymogens don't have T and R states as a result of the cutting of the peptide bond. We only see T and R state flips as a result of allosteric binding of effectors to the enzyme. Question 4 says, why does feedback inhibition not work with Zymogens? Well, this actually is rooted in the fact that feedback inhibition occurs as a result of binding of a molecule at the end of a metabolic pathway to the enzyme that catalyzes the first reaction. So let's imagine I've got a pathway of A going to B, going to C, going to D, going to E, going to F, for example. Okay? And let's imagine that this enzyme here, I'll just call E1, is an enzyme that's affected by the binding of the end product of the metabolic pathway. So this is what we call feedback inhibition. And feedback inhibition is an allosteric interaction. Again, the F is interacting with the enzyme, the very first enzyme, and causing it, in this case, to be inhibited. In the case of a Zymogen, we remember that Zymogens don't involve allosteric effectors, and Zymogens don't involve other molecules binding to them. Zymogens involve covalent modification of the enzyme, and this requires not a molecule, but rather another enzyme. So feedback inhibition cannot occur with a Zymogen simply because the Zymogen doesn't get affected by binding of other molecules. Problem 5 says, describe how ATCA's regulation is effective at measuring the pulse of a cell. Well, this also relates to something I talked about in class. Here's ATCAs, and we remember that ATCAs is affected by 3 molecules, 2 heterotropic effectors and 1 homotropic effector. The heterotropic effectors are ATP or CTP, and the homotropic effector is aspartate. So this is homotropic, and these 2 are heterotropic. All right? Now, for the purpose of answering the question, it doesn't really matter which one is heterotropic or homotropic. What matters is that the enzyme's activity is affected by each of these 3 compounds. ATP, for example, we can think of as an indicator of the energy state of a cell. High levels of ATP would be consistent with the cell having a high energy state. And as cells need to do things like divide, they need to have a fair amount of energy available to them so they can perform all the tasks necessary for the replication of the DNA and the division of the cell. CTP is a measure of the level of pyrimidine nucleotides. Well, pyrimidines, of course, are needed to make RNA and DNA. And if pyrimidine concentrations are high, the cell wants to turn off the enzyme, because by turning off the enzyme, it prevents the pyrimidine concentration from getting too high. That's not the case with ATP. ATP is an activator of the enzyme. And that's what we would expect if we wanted to have the enzyme become active when the energy situation was high. We also remember that ATP is a purine. So that when purines are higher than pyrimidines, we want to turn on this enzyme because this enzyme is necessary for starting the pathway to making pyrimidines. Therefore, activation by ATP makes very good sense. Inhibition by CTP, where the CTP concentration is too high, also makes very good sense. So between these two nucleotides, the cell can very easily tell what the status of nucleotides and energy is within it. The last molecule here is aspartate, and aspartate is an indicator of the amino acid status of a cell. If aspartate concentrations are high, then it's likely that the concentrations of amino acids in cells are also high. Therefore, high aspartate concentrations activate the enzyme because, again, the enzyme is going to need amino acids to make proteins in order to go through the process of cellular division. If the concentration of aspartate is low, we would expect that it would therefore, of course, not be able to activate the ATCAs. And that would make sense because when the amino acid concentrations are low, the cell wouldn't want to be committed to a process of cellular division.